Good morning. It's Monday, May 18th. Today we're going to talk about SLO 1 and 2. Your overview of those two SLOs should already be done. Today you're going to work on the notes and completing the analysis part of the DNA section of your webpage. If you have time, we've given you some extra links to play with on the agenda slides, and let's get started. First of all, we're going to start with the history of DNA. Back in the 1950s, scientists were trying to find this nuclein genetic material of the cells, and they knew that the cells were dividing and that they were communicating with other cells and organelles within the cell, and that when they divided, they needed to pass the information from one cell to the next through the generations, which means that you've inherited traits. The cells pass that information to the next generation cells so that the same cells get produced, the same jobs are done, and the species has the same abilities. This part of the cell they knew must hold a lot of information, especially for the more complex organisms and as many diversity of traits that we've seen across the organisms. The scientists knew that there had to be this genetic material, otherwise the molecule of life, that held all this information for these variation of the species. Watson and Crick were the two scientists credited for finding the structure of DNA. Rosalind Franklin was a physicist who helped also find that. She's one of the most famous women in science. And let's look at this DNA structure. The DNA, when it condenses into these tightly wound structures called chromosomes, that's the difference between DNA and chromosomes. But what is DNA made of? Most molecules are made of little building blocks, and the larger molecule is called a polymer. The building blocks are called monomers. For DNA, deoxyribonucleic acid, it's made of bases, adenine, thymine, guanine, and cytosine. The atomic structures are seen here. But it is in this polymer structure, and most of them are shown with this ribbon of a backbone, and then you'll see these rungs like a ladder, and the ladder itself is twisted in a double helix structure. The nucleotide, the monomer that makes up each of these, has a base. These are the four bases. It is attached to the ribose sugar and phosphate. It is the sugar and the phosphate that make this backbone. It is the bases that make the ladders. Now in RNA is ribonucleic acid. That's what, after the DNA creates a copy of itself and it sends it out to the cell to make proteins, the RNA carries the message to the ribosomes how to build those proteins. And we're going to study that in a few lessons. So don't worry about that yet. But what you do need to know is that the base in RNA, uracil, substitutes for thymine. Over here, you see the 23 pairs of chromosomes that humans have. Every organism, as you learned in previous lessons, has a different number of chromosomes. Humans get 23 from their father, 23 from their female. And if you want to practice building your own karyotype, it is a link on the website. It is also here, but you don't have access to these slides. So use the website for Science 8 to practice building your own human DNA karyotype. The karyotype word just means it is a picture of all the tightly condensed DNA called chromosomes shown, such as here. You'll also see them in cells, in drawings. They stain the nucleus to show that in regular inner phase, where the cell spends most of its time, the DNA is loose, it is not condensed. That's called chromatin, and you can see here that it's not visibly into the chromosome structure that you're familiar with from your mitosis and meiosis lesson. Right here, you see the condensed structures. They're drawn often in this structure here. This is what it looks like in real life. Now, central dogma is the term used for how DNA is in the nucleus, leaves the nucleus, gives a message to the cell to build a protein. So DNA, the word transcribed is what you need to know, means that the DNA strands that are double helix is copied into a single strand of mRNA. M stands for messenger RNA. And there are tRNAs in the cell, which means transfer RNA, which have little single strands that carry these amino acids, the building blocks of proteins, to build the proteins. As I said, you're going to focus on the translation in another lesson, but today I want you to just understand the structure of DNA. Now, in order to do that, you're going to do an activity around Chargaff's data. Chargaff's data is given to you on the website as a Google Sheet. You're going to make a copy of it, and then you're going to highlight the data and use down in the lower right-hand corner of the sheet, it'll show you the Explore button to create a graph. You've done this before in previous lessons. Once you get the graph, make sure you include a title, you label the axes, and then look for patterns. Watson and Crick worked with Chargaff, and he collected all this data around the bases. And he was looking for patterns and how this structure of the DNA might use adenine, guanine, cytosine, and thymine in those monomers of nucleotides to build the polymer of DNA. And that's what you're going to try and do. Look at the data, a graph will help you with that, and see if you can find patterns between these four bases. If you have any questions, as always, your teachers are available for you on Zoom. Have a great day.